So in the previous video, we talked about this phenomena of rancidity of oils, and one thing that is commonly done to overcome this problem is what's called hydrogenation of oils. All right, so most vegetable oils, all right, are going to contain unsaturated fatty acids, which are esterified to the glycerol molecule. All right, so as we mentioned before, these double bonds in these fatty acids are prone to getting oxidized, and that's going to allow for these triglycerides to become rancid. All right. So, and again, these two right here, if you want to draw in, it would just be other fatty acid chains. And one thing about these triacylglycerides is that these three fatty acids generally are not going to be the same. They can be the same, but like in any fat or oil, there's going to be some mixture of different types. All right. So anyways, in order to make these fatty acid chains a little bit more stable, one thing that is commonly done is hydrogenation. All right. And so if you treat this fat with hydrogen gas and a nickel catalyst, what you end up doing is turning some of these double bonds into single bonds. All right, and that happens because you're adding hydrogen to those carbon atoms. And when that happens, you end up getting rid of some of those double bonds. And so that process is called partially hydrogenating, or that creates partially hydrogenated oil. All right, and the reason they call it partially is that if you don't do it that long, you're going to end up getting rid of some of the double bonds, but not all of them. All right. If all of the double bonds were hydrogenated, you would say that it's fully hydrogenated oil. All right. So there's one issue with this, and that is, as part of this reaction, sometimes you get an unintended reaction. All right. So in just about all natural vegetable oils, the unsaturated fats tend to be in the cis configuration. All right, and again, if you put a line along that double bond, you'll notice that both of those bonds would be on one side of that bond. All right, so when and the thing about the cis fatty acids is those tend to be considered healthy or relatively healthy. But when you treat these under the same conditions as hydrogenation, you end up sometimes getting what's called an isomerization reaction, where that cis double bond gets turned into a trans double bond. All right, so this creates what's considered trans fat. All right, which is just a triglyceride, where some of these fatty acids have double bonds in the trans configuration. All right. And in general, the trans fats are considered to be not as healthy for you. All right, it's a fairly complicated science, and there's still a fair amount of debate about how safe these things are. But in general, the cis fats are considered to be more healthy than the trans fats. All right, and the interesting thing about it is trans fats aren't that really common in nature. They're made through a chemical reaction when you're trying to get rid of the double bonds. So what is the role of triglycerides to begin with, or fats and oils to begin with? All right, your body uses them as a very efficient way to store energy. All right, so if you take a look at the structure, you'll notice that most of this molecule is just a long hydrocarbon chain. All right, and if you notice on this part of it, there aren't any oxygen atoms. You would say that this is a very highly reduced molecule because it's holding just about as many hydrogen atoms as it can, and also there are no oxygen atoms in this part right here. So because this molecule is very highly reduced, if your body needed to turn it into energy, there would be a tremendous amount of energy that you could get from this. All right, because in general, organic molecules don't become any more energy rich than if they're just plain hydrocarbons. All right. So by comparison, another type of molecule that our body uses for energy are sugars or carbohydrates. Sugars are just a type of carbohydrate. But if you take a look at the structure, you'll notice that there are a lot of oxygens on this. All right. So pound for pound, if you had to burn a pound, if your body had to burn a pound of sugar or burn a pound of fat, you would get more energy from burning the fat, just because the types of molecule, the types of atoms that it has in it, would give off more energy when they get oxidized, right? All right. And if you've ever watched a documentary that talks about, like, people doing like polar expeditions, all right, whenever people are doing any type of activity where, first of all, you don't know where your next meal is going to come from, and while you're doing it, you're going to burn a lot of calories. And if you're doing a polar expedition, your body's going to expend a lot of energy just trying to keep its body temperature regulated. It's to your advantage to eat things that have as much energy as possible. By the way, that's Mars back there. All right. Anyways, I just wanted to point out that in addition to a very, very efficient form of energy storage, the second is that it also functions as insulation for the body. Okay, so we're moving on to this another type of lipid called a phospholipid. All right. In general, you would not consider a phospholipid to be fat, even though it's chemically similar to a fat. All right. So whenever you have a true fat, you're gonna have a glycerol molecule which has been esterified to 3 separate fatty acids. All right. So with a phospholipid, essentially this middle fatty acid is removed, and it's turned into this piece right here. All right. So I've shown it on this side right here for a reason, but this is, again, this middle part right here would just be a glycerol molecule. You've got the same esterified fatty acids on the 1 and the 3 hydroxyl of the glycerol molecule, but in that center one, you've got you have this little piece right here. This is a phosphate group, and it's got this part right here, and that part of the molecule is called choline. All right. So in general, they call these this whole molecule a phosphatidylcholine. All right, you're not responsible for knowing that, but these phospholipids, and this is I guess a generic example of one, play a very, very important role because they make up our cell membranes. All right, so I've kind of tried to take this drawing right here and shrink it down a little bit. Hopefully you'd see that. Okay, if this is a fatty acid, it would be a really long chain. All right, so this entire thing right here would be like this entire thing right here. And the one thing about these phospholipids is that this part of it right over here really likes water. All right, there's actually a charge on that nitrogen, and whenever you have charges, water really likes it because water is a polar molecule. But this side of the molecule over here really doesn't like water. All right, so this part of the molecule is hydrophobic. This part right here is hydrophilic. All right, and so if we put these, if we start stacking some of these together, what's going to end up happening is that these long chains right here are going to tend to like to stick to each other. All right, so if you put them together, you end up getting it so that, all right, well, these guys are going to stick to each other, but you're going to have this other, an opposing side that also likes to stack together like this. All right, and this is going to be essentially what a cell membrane is made of. All right, so just to give you a little sense, like if this is the cell membrane, picture this thing going around into a big circle. Actually, the big circle would go around this way, and inside the cell you've got some water. All right, and this part of the phospholipid, since it's got that charged nitrogen, really likes that the water from the inside of the cell. All right, and on the outside of the cell you also have water. All right, so again, I've kind of I haven't drawn the whole thing, but draw in oxygen, and this part right here on the outside, that also likes water. All right, so The thing on the inside is really hydrophobic. The interior of the cell membrane really doesn't like water. All right, so essentially that makes a really good way of separating the water that's inside the cell from the water that's outside the cell, because there's no way that a water molecule would like to go through this. All right, it's a very uninviting environment right here for water. Water would very much like to stay around itself or to this part of the phospholipid molecule. All right, so the last type of lipid, which is almost its own separate class, is steroids. All right, and one thing that about steroids is that they all have this same general ring structure. We call this a 6-6-6-5 ring structure, and this makes what's called the ABCD fused ring system of a steroid. All right. So there's two major functions of steroids. Is that first of all, they are components in cell walls. The classic steroid for that is cholesterol. But they're also used as signaling molecules, and the classic molecules for that you can think of testosterone and estrogen. All right, so this right here shows the structure of cholesterol. All right, and you notice it's got that 6-6-6-5 ring system of steroids, and you can always spot a steroid fairly quickly because it's going to have that general structure. Okay, and so structurally similar to that is testosterone. All right, and the interesting thing is they have a very clear structural similarity. You have that 6-6-6-5 ring system, but they serve totally different purposes. Testosterone serves as a hormone in the human body that triggers a lot of physiological change when especially boys go through puberty. But cholesterol is something that's used primarily in cell membranes. All right. So the reason that steroids are considered lipids is that the thing that makes all lipids similar is that they tend not to like water, and they tend to be very soluble in more nonpolar things. All right. And so if you take a look at the molecule, you'll notice there's lots of carbons and hydrogens, not a whole lot of polar bonds. All right. And so if you were to take cholesterol and try to mix it with water, you would notice that they don't mix that well. All right, so that's what makes it considered a lipid. All right, so stepping back, I wanted to show a quick graphic of the phospholipid bilayer that we talked about a second ago. Okay, so I know that this looks kind of busy, but like the glycerol part of this molecule. All right, so this part of the molecule would be right here. All right, notice that you've got a regular old fatty acid on the 1 carbon, and you also have it. This one actually shows it on the 1 and the 2. You can have it the phosphate on different alcohol parts of the glycerol molecule. It can either be on the 1 and the 3 or on the 1 and the 2. But the important thing is that 2 of these are going to have fatty acids, and one of them is going to have that polar phosphate group. All right. So this right here shows this, and this would be the long chain fatty acid, and this right here is going to be that phosphate group, this part of the molecule right here. All right, so this part right here is the part that really likes water. All right, that would be this part right here. And these parts right here are the fatty acid chains, and those don't like water. All right, so if you take a bunch of these right here, a lot of times they represent it like this, where the circles here are the hydrophilic head. All right, and they're going to stack next to each other like this. All right, so you're going to get 2 layers of them. That's why they call it a bilayer. All right, and so one side of this would be the inside of a cell, and the other side would be the outside of a cell. All right, and one thing that you'll notice here, you see what looks like a 6-6-6-5. That's actually a molecule of cholesterol that gets embedded in the cell wall, and that helps to it serves a structural function within the cell membrane.